Okay, while I set up, may I get your help to stand by also? Round 2 of games are coming up. Since you won, right? There you go. Don't hurt yourself. Yeah, I'll send the next round of photos today, so you'll stand by. I saw something in the marina area that's quite interesting. Yeah, how about we play now? Okay, I'm going to send a video now. The game is guess the location in Singapore. Guess the location in Singapore. No, it's not geography, it's biology. Yeah, I'm sending this to you right now. It's a video. Your Wi-Fi link is not loaded like that. I'm sorry. Okay, sorry, you say cannot send a message. Anti-connects. Again, cloud Wi-Fi connects. Guess not. Perhaps after. Okay. Today, we're going to finish off our last chapter for the term. I'd like you to take out your notes for photosynthesis. The last round, we stopped at this particular part. Right, sorry, the one below. How chlorophyll is able to capture and use energy. expect you to give a quick read of page 6. Can I get you to give page 6 a quick read? Now in page 6. Okay, once you're done in page 6, please read page 8. Okay. last week? Was it last week? Last week we learned that chlorophyll can absorb light energy, right? And when they absorb light energy, something quite special happens. The electrons get excited, right? On the whiteboard, here I have two large structures. These structures are called, I don't know if you remember the name. Starts with a P. Thank you. Photosystem. Okay? It is a collection of a lot of proteins there. We also say that photosystems are where you can find lots of chlorophyll with electrons that are excitable. Okay? So what I have over here, each circle represents a chlorophyll pigment. Each yellow magnet represents electrons that can be excited. Okay? We learned also that photosystems collect light energy in a certain way. When it gets struck with light energy, the electrons can get excited. Okay, so I'm going to represent excitation by it popping out of the whiteboard. Okay, ready? So light strikes the photosystem, the electrons of the chlorophyll pigment get excited. But everything that gets excited needs to be unexcited after a while. So it goes back down to its normal state. But as it's going back down, it passes its energy that's releasing to the next electrons to get excited. And almost like a wave that you sometimes do at a football match or a game, you know, we just pass the energy on to the next one and to the next one. So that's essentially how our photosystems with a lot of chlorophyll pigment transfer energy. Transfer energy through the excitation and de-excitation of electrons. Today we're going to see how all this relates to creating ATP. Okay? You can see that actually this was very familiar. Doesn't this look quite like the electron transport chain in the cellular respiration? The very last step of oxidative phosphorylation. Very, very similar. So if you understand how it works for oxidative phosphorylation, this is very similar. The biggest difference is where they are pumping the protons through. What is used as who is donating the electrons, who is receiving the electrons at the end. As we go through this, I hope you bear in mind and slowly can make comparisons to oxidative phosphorylation. Because I could ask you, can you compare the electron transport chain found in the mitochondria and in the chloroplast? And you should be able to compare and contrast. I will list out as many comparison points today so that we can see some of them. Okay? So, I want you to pay attention to this particular electron over here. Remember that after they pass the energy down to this electron right here in the special chlorophyll, it gets so excited by all the energy transferred to the middle that it kind of jumps out of the chlorophyll and up into this primary electron acceptor. And from there, it jumps to the next protein in the electron transport chain. Now, just like normal electron transport chain, electrons will flow along the chain. We're going to watch a video now to see how this entire process happens. Okay, we get an overview, then we talk about it a little bit more. Same thing, interactions. Who is involved? What changes? Where does everything take place? So for a start, this all takes place on the thylakoid membrane. Okay, this is over here. It's a thylakoid membrane. Which are those discs on the membrane itself. Okay.
Plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma.
Okay, so that's a visualization of how this process runs. Notice there are some similarities to oxidative phosphorylation. Yeah? Okay. So let's do a quick recap of what you saw in the video. Okay, it's early morning. If you want to drink some water, go wash your face, please do so. Yeah. Okay, so here we have a thylakoid membrane. We have photosystems. Alright, I'm going to call this one over here photosystem 2. And this is photosystem 1. You may be wondering why I call this one number 2, this one number 1. Because the scientists discovered this first. So they thought this was the only one. Until they discovered this one and they named it after. So that's why the odd naming convention here. We learned earlier that light energy strikes both photosystems at one go. So light energy also strikes this photosystem here. So follow me right now. What will happen if light strikes both photosystems? All the electrons will get excited. Except I don't have enough hands. Okay, so may I get the help? So, you will help me with photosystem 1. I will do photosystem. Okay, so you do photosystem 2, I'll do photosystem 1. Okay, light energy strikes. So let's excite the electrons. Ready? Okay, excite. Okay, pass energy to the next electron. Pass energy to the next electron. Into the next one. And to the final one. And it jumps up. Goes to the primary electron acceptor. And now it passes on down the electron transport chain. Okay? Alright. You continue. You go down the electron transport chain. Continue on. Okay? Okay, see a problem here? If light continues to excite these electrons, energy will be passed along, but passed along to nobody. This over here now is missing electrons, right? Where can we replace electrons from? From just next door. Okay, so you go in. So now I can continue the process. Light strikes, excite, 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 and then jump out, and we can continue the process on, right? Okay, but then we have a problem here. We are missing electrons here, right? Where can we get the electrons from? Okay, so in your notes and in the video, the electrons come from water. Notice it's kind of the reverse. In oxidative phosphorylation, we give electrons to oxygen and we form water. Now we are taking electrons from water. Okay, let's take electrons from water, and as a result, we end up getting oxygen that's being produced. Yeah? So kind of the reverse. And then we can continue on, right? Okay, thank you. You can go back again. Okay, so, but where do all the electrons go? Right? When we look at oxidative phosphorylation, where did the electrons come from? They came from NADH, remember? But now we're doing the reverse. We're going to pass all the electrons to something very similar called NADP+. Okay, this is plant's version of NAD+. Okay? When NAD+ gets electrons, it becomes NADPH. So really, it's kind of like the reverse. We were passing the electrons one way in oxidative phosphorylation. Now we are passing it the other way. Yeah? As the electrons flow down the chain, the usual stuff happens. Energy is released and we get to pump hydrogen ions. Except we pump it kind of a different direction now. In oxidative, I kept pumping it out from the matrix to the intermembrane space. In this case, we pump it inside. Okay? So I'm going to pump all of these fellas into the thylakoid space. Setting up what we call a proton gradient. Where there's high H plus in the inside and very low H plus in the stroma. Same thing, we are just setting up proton gradient. So if the proton gradient is in this way, how will the protons flow? From the intermembrane space into the, what am I talking? From the thylakoid space to the stroma. Right? Okay, so let's flow. And as it flows through, it drives the ATP synthase to produce ATP. So just a quick practice for yourself, since we're looking at interactions. Can you write down what is needed? What is produced? Okay. What is needed? What is produced? just ask some of you on the floor. Nicole, what is needed? What is needed for this entire thing to work? Water. Okay, we need water. Did everyone get water? Okay, water is one of the things we need. Thank you. Where is it needed? Or what is it needed for? Okay, this is the vocab you could use. We need it as an electron donor. Something that donates electrons. And if you read your textbook, how does it donate the electrons? Through a process called photolysis. Photolysis means we use light to break what? Break water. Thank you, Nicole. Anything else needed? If you look at the whiteboard or record a video or you read a textbook, what else is needed other than H2O? Hai? Light. Okay, fair. We need light because without light we cannot excite this electron in a special particle. Fair. We need light. Anything else we need? Yes. NADP plus. NADP plus. Okay, as an electron acceptor. Okay, so the vocab you could use is the NADP plus can receive the electrons, right? Okay, we refer to NADP plus if that's the electron donor. This is a electron acceptor. In fact, you can say it is a final electron acceptor. Who's the final electron acceptor in oxidative phosphorylation? Oxygen. Can you see some points of comparison now? Okay. Can you tell me therefore who's the electron donor in oxidative phosphorylation? Who donates the electrons to the electron transport chain? Can you remember? NADH. Okay, I'm going to write down what you can compare. So, in the context of oxidative phosphorylation, the electron donor is actually NADH and NADH2. But in the context of what we call this stage is the light dependent stage. We actually use H2O. Okay, so there's one thing you can compare. As the final electron acceptor in oxidative phosphorylation, it is oxygen. But in the case of the light dependent stage, it is NADP plus. Here we just compared two things. Okay, another thing you can compare, I'd like you to try to write this down yourself. How will you compare the direction in which the photons are pumped? Okay? So, point of comparison number 3. Photon pumping. In the case of oxidative phosphorylation, is from where to where? In the case of this particular light dependent stage, it's from where to where? another thing to compare. Which membrane is the electron transport chain on? For oxidative phosphorylation, it is where? In this particular case, where is it at? Okay, then we will go through some of these points of comparison, but you try yourself first. Hard to say opposite now, because they are different locations. So how will you describe from where to where? Yeah, just try to remember. If not, you can refer to your previous set of notes. Once you think you've got the comparison point for proton pumping and membrane, you share with your friend, then I will not tell you the answer. I'll only share a few things you can compare. I'd like you to compare yourself, yeah? If I come out during exam, then they say I help this class. Yes. So I just tell you what you can compare. And one last point of comparison that Jing can help with, point number 5, is light energy needed. Okay, for oxidative phosphorylation, do you need? For light dependent stage, do you need? Yes. Oh, where is the electron transport chain found? Which membrane? Yeah? Okay, what? We're going to write here. Where is the ETC found? Okay, you should tell me which membrane. comparison, this one a bit lame, is related to light energy. See some students compare and contrast whether chlorophyll is needed. Or sometimes they will compare whether photosystem are needed. Okay, but these are very similar points. It's a stroma. What things there's anything else to compare? I think I maxed out what I can think of. Anything else you think you can compare? Then we help each other out now. Oh, okay. Where does this entire process take place? Okay, you can talk about the organelle. This is the chloroplast after all, right? The other one is the mitochondria. I hope you have enough points to form a table of sorts. Okay, but with every compare and contrast question, there are differences and there are similarities. So what are the similarities? Okay, let's circle similarity number 1. There is ATP synthase. Okay, any other similarities? Okay, yeah. Number 2, if we look at the entire thing, in number 2, both of them have electron transport chain. Anything else? I thought proton gradient, is it? Okay. Number 3, you will set up a proton gradient. Anything else? That's about it, Okay, maybe make it a bit clearer. Okay, so these are similarity 1, similarity 2, similarity 3, differences. I'll leave this on the whiteboard for you to touch up your notes. You want, you still can snap a photo and put inside your notes also. Today we have a guest speaker during CCE. So I invited this company to share about their prototype. They made edible spoons. So you can, after you use it, you can eat it. Yeah? I've been talking to them since like last year, and actually they went through many prototypes. Their first prototype was a certain way. They are getting like prototype, I don't know, number 1. So later on you get to see their product. There are a few flavors inside that are ordered. You have to fight for it. There's like burnt caramel, then there's like banana goji, and there's strawberry something. It's strawberry basil, strawberry basil, whatever. Yeah. Yeah, the spoon is biodegradable. So this flavor is brown caramel oats. Makes everyday food more nutritious. Yeah, crunch cutlery. Okay, our class has gotten banana goji as well as caramel oats. Last year they were like asking me, to like give feedback and vote. Which flavor do you think I want more? Then I said, banana goji sounds like a nice flavor. Yay! So they created banana goji. It's not the same goji effect. Goji is the red berry, right? Okay, so we are nearly towards the end of photosynthesis. We have not talked about what is produced. So quick one up. Wow, Actually, I got shocked. The champion put down his mask. I was like, oh, champion looks like that. You know, I mentally fill in the blanks what's below. Like, I don't quite know what you all look like, you know. So, okay. So, what is produced? Two things are produced, right? One thing that's produced is ATP. Another thing that's produced is NADH. But for what? NADPH. Okay, these are our two products of the light-dependent stage. Now, what are we using this for? Okay, it's for the next step. The next step, we call it the, okay, good news for you, photosynthesis only got two stages, light-dependent and light-independent. Okay, so everything will be channeled to the next step, called the light-independent stage. This means that the next stage does not require light. Good. And that also means that I can technically supply a plant with light, then I switch it off, the next stage can actually still run. Okay, the plant will not die immediately. Okay, let's go to the next stage, the light independent stage. Yeah, right? If you look at this particular stage, doesn't it look like the stage right before oxidative phosphorylation? Remember that cycle? Krebs cycle? In Krebs cycle, what were we doing? We were pulling carbon dioxide out, right? Remember? Other than pulling carbon dioxide out, we were also generating a little bit of energy in the process. Here, we're also pulling electrons out, remember, from Krebs cycle? I know what we're doing here? We do the reverse. Let's take carbon dioxide. Let's take the electrons from NADH. Let's take the energy from ATP, and let's build sugars. So plants do the reverse. It takes whatever electrons it has gathered, it uses the energy that in the form of ATP to eventually produce sugar. Do I need you to know every single step of this cycle? No. Okay, so what should we know? Interactions. Just need you to know what is needed, what is produced. So a quick one for all of us here. Just looking at this cycle, you can also read the parts below. Can you tell me what is needed for the light independent stage? Okay, so let's list down some of the points. I'm going to erase the photon gradient away. Ready? Okay, what is needed for the light independent stage? Number one, CO2. Okay? Now, I tell you, if you ever forget what's needed, what is produced, right? You go and write down the photosynthesis equation. You need to fit it in somewhere after all. Right? What else is needed? Okay, whatever produced in the light dependent stage will be needed. Okay? Because it's kind of the reverse Krebs cycle. Okay, so NADPH will be needed. You also require ATP. These three things look very similar to the products of Krebs cycle. Right, don't you think? We are just really going in the opposite way. Wouldn't it be great when photosynthesize? Are you the class that asked when we photosynthesize? Because then we were discussing, if we can photosynthesize, right, then where should the chlorophyll be? It should be on our skin. Right, then we run around. Yeah, we'll look green. But then you see, all of us wear clothes. If we're not maximizing the surface area, we have to run around naked, right, to fully maximize the sun. That's true. But also, right, the surface area to volume ratio doesn't make sense. We have a lot of parts inside us, right, that won't touch the sun. So the only way, right, is that we need some part of our body to maximize surface area, right? Plants have leaves. So I was thinking to myself, maybe what we need, right, is like green hair. There's lots of hair, and we need really long green hair that stands out. Then everybody just run around with one pot of like grass on the head. That's the only way I can think of, right? Where else can maximum surface area on your body. More limbs, like cannot be, right? Okay, sorry, I keep diluting. So what is produced? What is produced from the light dependent stage? Okay, for those that are reading, what is produced is this sugar that's made out of just 3 carbons. Okay, so we will say it's a 3 carbon sugar. Okay, for your information, we need 2 of these 3 carbon sugars. Okay, times 2 equals a 6 carbon glucose. This means we actually need to run this Kelvin cycle a few times before we can actually get enough 3 carbon sugars to piece together and make glucose. Are there any more products of Kelvin cycle? Which is related to the top up. If we use these 2, what will we get as the byproduct? ATP and NADP plus, right? Yeah. Notice, that's why the idea of what is produced and what changed is the same, it's synonymous. This carbon dioxide will become our 3 carbon sugar. This NADP will become our NADP plus again, after it gives up its electrons. This ATP will become ADP again. I will pause now to take questions. Maybe I've been going too fast. By now we've realized that this is very similar to cellular respiration, kind of just the reverse, right? Therefore, for you, what's the best way to study these 2 chapters? Sorry? Okay, actually, the best way would be to compare and contrast. If you can compare and contrast the 2 processes, you would inevitably revise both at once, and you prepare yourself for a question that makes us do together. Okay? After all, we don't call this chapter cellular respiration or photosynthesis. We call it energetics. Yeah? Because this is the study of how organisms produce energy, make use of energy. That's why it's called energetics. Another tip when studying this chapter is, there are too many processes written down in words. I encourage you to watch videos on Lumos. Visualize the process. Don't look at the words and try to remember the words. Visualize the process, remember the process, and then you describe the process from your mind. Yeah? I often find memorizing the steps not very useful because it doesn't give justice to the movements here and there. Like when I say protons are pumped, right? You memorize the sentence versus you remember how it goes, right? It's a very different feeling. That's why we also went through the role-play, right? So that we can experience the process ourselves. If we be it, we will remember it. Okay, around 10 more minutes. I'd like to draw your attention to these 2 graphs. Unfortunately for us, we've only touched on one kind of chlorophyll. There are actually many kinds of chlorophyll. The chlorophyll we are most familiar with, the ones we see in our plants, green. Actually, have you seen plants that are of different color? Alright? So chlorophyll does come in various colors. The various colors you see simply implies that this chlorophyll can absorb different wavelengths of light. We actually have chlorophyll, they are purple in color. Yeah? So here we provided you three kinds of chlorophylls. Chlorophyll A, chlorophyll B, carotenoids. Below you see the wavelengths of light. The graph above is simply telling you for every chlorophyll, what range of light does it like to absorb? If we look at chlorophyll A, will you tell me which wavelengths of light it likes to absorb? What's the range? Okay, so expect that this may come out as like a graph question. Then we ask you, chlorophyll A absorbs light in which spectrum? Can you tell? Okay, or maybe another one. Chlorophyll A's peak absorption is at which spectrum of light? Around 430-ish, yeah? What's the second peak? 660-ish. Okay, so if you ever see this graph, you have to draw dotted lines to properly estimate. Okay? Plants have more than one kind of chlorophyll. These are the more common ones, A, B, and carotenoids. If we compare it to this second graph below, this graph shows the rate of photosynthesis. Notice that the rate of photosynthesis corresponds to the wavelengths of light that the chlorophyll absorbs. Whether it's A, B, or carotenoids, the absorption tends to be between 400 to 500, 600 to 700. And it corresponds to the rate of photosynthesis. We find that if we supply plants with light from this range and this range, the rate of photosynthesis becomes higher. Right? So that's what these two graphs mean. That's why we do not supply plants with green light. Right? Actually, if we take a plant, we just shine it with green light, right? It's not going to survive very well. Have you ever been to the agri-tech before? Yeah, I don't know if you've seen this before. Sometimes at hydroponic or some factories, when they want to grow plants indoors, they don't supply it with like white light. They instead supply it with purple light. Right? That's all they supply with. Like, don't waste energy. Don't waste energy supplying all spectrums of light. All they supply is purple. The plants grow really, really, really well. They omit the green effect. So actually it's very interesting. If you ever were to see these plants, right? If I don't supply green light, will my plants still look green? After all, the green color we see on the plants is just a reflection of what was not absorbed, right? So if I only supply purple, which is blue and red, no green at all. Actually, when I step into and look at these plants, right, these plants don't look green at all. They look like an odd, very dark, gray kind of color because it's absorbing everything that we are supplying. So it's quite interesting phenomenon. Which also means, right, it's very interesting. Okay, then I ask you, actually, when in darkness, do we see a color? If after all, what the colors we see are simply a reflection of what's left over, not absorbed, and therefore, actually, in black, right, all of us have no color. Right? Yeah, right? So, actually, in complete darkness, it's not that you cannot see, it's just that we have no color. It is physics, I think. Who's your previous teacher? Mr. Lin Ching. Okay, we have come to the end of photosynthesis. So expect that when we come back from the March holidays, you will have your first quiz. This quiz is ungraded, but it's a fantastic opportunity to test your notes. See if your notes work. We've already put up an announcement on Google Classroom on the instructions of what you can do or can't do for your notes. Okay? as I check through your submissions, I have a lot of overdue submissions. A lot of you have not submitted work or corrections. Okay, I'm going to compile Excel. This is what I normally do at the end of each term. Okay, because at the end of each term is when I key in all the grades. Then I'll, hey, can switch off the iPad.